Hello and hello everyone, welcome to class 12 biology. With this video we are going to start a new unit from our biology textbook, that is biotechnology. And the first chapter of that particular unit is chapter number 11, biotechnology, its principles and processes. So we will start with the definition of what is biotechnology. So biotechnology can be defined as use of genetically modified organisms or GMOs to produce products and processes that are useful to human beings. So that is the conventional definition of biotechnology, but the European Federation of Biotechnology, or EFB, has defined biotechnology as the integration of natural science and organisms, cells, parts thereof, and molecular analogs for products and services for the human beings, right? So that is the definition given by European Federation of Biotechnology. So for your examination purpose, you should remember both these definitions. Right, definition of biotechnology in a conventional sense and the definition given by European Federation of Biotechnology. The two core techniques that is said to have led to the birth of modern biotechnology are genetic engineering and bioprocess engineering. So what do we mean by genetic engineering and bioprocess engineering? By term genetic engineering, we mean alteration of genetic materials like DNA and RNA and introduce those changed or altered DNA and RNA into host organisms, thereby changing the phenotype of the host organisms, which means the creation of genetically modified organisms. Right? We create genetically modified organisms by first making alterations to their genetic material, right? Let's say DNA. So we make some changes in that particular DNA, and that we introduce that modified DNA into a particular host organism, and due to that, right, the host organism's phenotype changes, and therefore that host organism is called as genetically modified organism. So such technique is called as genetic engineering. So that is one of the core technique which led to the birth of modern biotechnology. Now, apart from that, we also have got bioprocess engineering, in which, right, maintenance of sterile ambience or maintenance of sterile environment was made possible. So this grew in chemical engineering field. Right. Before coming into biological field, it first started in chemical engineering field, in which the scientists, they were able to maintain sterile environment for their experiments. Right. What do we mean by sterile environment? We mean contamination free zones. For example, these two scientists, they are working in a particular hood, right? This particular hood is called as laminar flow. Okay, so this particular table or this bench like structure is called as laminar flow, in which on up top there are ultraviolet radiations. Ultraviolet radiations will first of all kill all the microbes that are present on this particular surface, on the surface of this table, and then there is filtered air blowing from the front of these two scientists. The filtered air will be blowing from the front of these two scientists, which will prevent the entry of any unwanted spores of bacteria or fungi coming into the table in this particular environment. And not only that, all the gloves, all the equipments, all the glass slides, petri dishes, everything will be sterilized before being brought into this particular table, right? So such kind of techniques where a sterile environment is maintained, right, that is called as bioprocess engineering. And that this particular process, when it was adapted into biological field, it made the biotechnologist or the people who are working with microbes, they can grow only those microbes, and no other contaminants like other bacteria and fungi will grow on their petri dish, right? Or if a scientist is trying to grow a particular cell, let's say a liver cell, right, so he can grow only liver cell in the petri dish, and no other contaminant will grow in that particular petri dish. Right? So such technique is called as bioprocess engineering, and that is the second core technique which led to the birth of our modern biotechnology. So these are the two core techniques which led to the birth of modern biotechnology. Now let us try to understand the term genetic engineering in a better way by looking at an example. In this particular picture, you can see a petri dish with bacterial colonies growing in it. Right. So this petri dish contains nutrient medium, and in this nutrient medium there are lots of bacterial colonies growing in it. Now if you expose this petri dish and the bacteria to normal light, under normal light, the bacterial colonies, they do not glow. But if you expose the same petri dish under UV light, some of the bacterial colonies, they have started to glow. Right. So these bright green spots, they are glowing under UV light. They are bacterial colonies which are glowing under UV light. Now, why are these bacterial colonies glowing under UV light? That is because the bacteria in these particular colonies, they are producing a particular protein called green fluorescent protein, or GFP in short. The green fluorescent protein is the protein which has the capacity to glow under UV light. But the bacteria, the original bacteria, they do not have the gene necessary to produce green fluorescent protein. Right? Green fluorescent protein gene is not part of bacterial genome. These bacterial colonies, the bacteria in these particular colonies, they are glowing because the scientists, they have introduced the gene for green fluorescent protein into bacterial genome, and therefore they are producing green fluorescent protein, and therefore they are glowing under UV light. Now, where did the scientists they get the gene from, the gene for green fluorescent protein? The scientists got the gene for green fluorescent protein from marine organisms like jellyfish, deep sea jellyfish. These deep sea jellyfish, they naturally contain the gene for green fluorescent protein, and they glow under dark marine environment. Scientists, what they did, they identified the gene for green fluorescent protein, they extracted the gene, and somehow they introduced the gene into the bacterial genome, thereby making the bacteria capable of producing green fluorescent protein, and therefore these bacterial colonies, they are glowing under UV light. Now, this is called as genetic engineering. The scientists, they somehow created a genetically modified bacteria over here. So the genetically modified organism over here is the bacteria which are glowing under UV light, right? So this is genetic engineering, but can we call this as biotechnology? What is the benefit of a glowing bacteria under UV light, right? So let's imagine another way, right? Instead of taking gene for green fluorescent protein, if we take gene of human insulin, if we take the gene for human insulin from the human being and insert it into the bacteria, what will the bacteria do? They will start producing human insulin hormone, and we can take that human insulin hormone and inject into diabetic patients. That is useful for the human beings, right? So that is biotechnology. So genetic engineering is one of the important processes in biotechnology. Now, genetic engineering involves certain important techniques. Those important techniques of genetic engineering are: first, creation of recombinant DNA. Second is gene cloning. And the third is gene transfer. Let us try to look at creation of recombinant DNA. How do we create a recombinant DNA? If we look at the history of creation of artificial recombinant DNA, two names will come first, right? So those two names will be Herbert Boyer and Stanley Cohen. Herbert Boyer and Stanley Cohen were the first scientists who were able to transplant genes from one organism to another. Right? We have heard about organ transplants, right? We have heard about kidney transplants, heart transplants. But these two scientists, Herbert Boyer and Stanley Cohen, these were the first two scientists who were able to transplant genes from one organism to another, which was a fundamental discovery for genetical engineering. Okay, so in 1972, these two scientists, they managed to make bacteria antibiotic resistant. How did they make a bacteria antibiotic resistant? They made a bacteria antibiotic resistant by linking antibiotic resistant genes with the plasmid of Salmonella typhimurium. We will try to understand plasmid, more about plasmid later on. But what they did was they took a plasmid from Salmonella typhimurium and they inserted the gene responsible for making a bacteria antibiotic resistant. They attached this particular plasmid with the gene and they created the first recombinant DNA. Right. So these two scientists were the first to create an artificial recombinant DNA. Now this circular DNA over here is called as recombinant DNA because it contains DNA from two different organisms. Right. Remember, these two scientists, they took this particular plasmid DNA from Salmonella typhimurium bacteria, whereas they took this antibiotic resistant gene from some other bacterium. And therefore, the combination of these two DNA resulted in recombinant DNA. Now, they were able to make bacteria antibiotic resistant by introducing these recombinant DNA into those bacterium. Now, if we say we have a bacteria which is not antibiotic resistant, if this particular recombinant DNA is inserted into that in this particular bacterium, the bacterium will now become antibiotic resistant because it now contains the gene responsible for antibiotic resistance. Okay, now let us see what are plasmids. Plasmids are autonomously replicating circular extra chromosomal DNA found in bacterial cells. Inside the bacterial cell, there is the main bacterial DNA or the main bacterial chromosome. So this red one over here is the main bacterial chromosome. Apart from the main bacterial chromosome, there are extra chromosomal small circular DNAs called plasmids. And these small circular DNA, they can replicate autonomously, which means the replication of these plasmids does not have to depend upon the replication of main bacterial DNA. You can look in this particular picture. When the bacterial cell divides into two bacteria, what happens? The bacterial DNA, it replicates and divides itself into these two cells. Along with that, the plasmids, they will also replicate by themselves and divide themselves into these two cells. So these are plasmids, and plasmids are pretty important because they act as vectors. You know what are vectors, right? When we discussed about disease causing organisms, when we discussed about malaria, I said that mosquitoes are vectors because mosquitoes are the ones which carries the disease causing organism or the pathogen called plasmodium. So vectors are the ones which carry a certain kind of disease causing pathogens. Just like that, these plasmids, they are also called as vectors in our biotechnology or in genetic engineering process, because plasmids, they have got the capacity to carry foreign genes into them. So these plasmids, they can carry foreign genes into some other organisms. So therefore, these plasmids, they are important molecular tools in genetic engineering. Okay, let us now try to understand the basics of genetic engineering. Like I told you before, genetic engineering involves three important techniques, that is creation of recombinant DNA, gene cloning, and gene transfer. These are the three important techniques involved in genetic engineering. With the help of this particular diagram, let us try to understand these three techniques. Okay, so over here we have a human cell and a bacterial cell. From the human cell, we take out a gene of interest. In this particular case, we have human insulin gene. So we take out human insulin gene from the human cell, and from the bacterial cell, we take out the plasmid DNA or the vector. And then we combine these two DNA to create a recombinant DNA. Now remember, for the creation of recombinant DNA, we require a very important biological tool called restriction enzymes or restriction endonucleases. Restriction enzymes or restriction endonucleases, they are enzymes which can cut DNA at specific locations. Right? Restriction enzymes are like molecular scissors. They can cut DNA at very specific locations. Over here, we have used a specific restriction enzyme in isolating human insulin gene from the entire human genome. And when we use restriction enzyme, the restriction enzyme cuts the DNA in a manner in which it creates sticky ends at the opposite corner, right? Sticky ends are nothing but single-stranded DNA protruding out of the double-stranded DNA. And these are called as sticky ends because whenever they come across a complementary DNA single-stranded DNA, it tends to stick with that particular DNA single strand. Therefore, they are called as sticky ends. Now, if we use the same restriction enzyme on the plasmid DNA will also be cut, and on the plasmid DNA also we will find sticky ends. And the thing is, since we used same restriction enzyme on both these DNA, we get complementary sticky ends, and therefore these two DNA will stick with each other. And by use of DNA ligase, we can join these two DNA and form or create a recombinant DNA. So we have the recombinant DNA over here containing the human insulin gene. Now this recombinant DNA can be inserted into a bacterial cell or a bacterial host cell. And now this bacterial host cell will be a genetically modified organism because this bacterial cell now contains human insulin gene in it. Right? And this bacterial cell will start producing human insulin gene. Now over here we have seen the creation of recombinant DNA. Now let us try to see gene cloning and gene transfer. Now in gene cloning and gene transfer, whenever this bacterial cell divides to create progeny bacteria, along with the bacterial chromosome, this particular plasmid DNA or the recombinant DNA will also replicate. I told you, plasmids, they can replicate autonomously. And whenever the bacterial cell divides, the plasmid DNA or the recombinant DNA will also replicate, thereby creating clones of itself or copies of itself along with the human gene. So the human gene of interest or the human insulin gene will create copies of itself. That is called as cloning, right? And the gene will be transferred to the progeny bacterial cells. Okay, so with this diagram we can see all these three techniques involved. So that is the basics of genetic engineering. And in short, three basic steps in genetically modifying an organism are identification of DNA with desirable genes. In this particular case, the gene of interest is human insulin gene. We identified that particular gene, and then we introduce the identified gene into the host. This is the bacterial host. So we introduce the human insulin gene by use of a particular vector or a plasmid DNA by creating this recombinant DNA, and then we introduce this human insulin gene into the bacterial cell. And then we maintenance of introduced DNA in the host and transfer of DNA to its progeny. Since this bacterial DNA or this bacterial cell, whenever it divides, it carries a copy of this particular human gene. The human insulin gene is maintained in the progeny and it is transferred from progeny to progeny, okay, from bacteria to bacteria. So that is the basics of genetic engineering. In the next video, we will discuss about tools of recombinant DNA technology. So you should read from section 11.2 onwards, and then you can watch the next video. Thank you.